should do the thumbnail. It should be like that.
Oh, the thumbnail? I think we already got it. We got it. But let's just keep on trying. What do you want? Just look into the abyss and say, this is it.
You say like, oh, nice jumper, eh?
Okay. Okay, perfect. Yeah. All right. Well, I'm happy you like it, and yeah, you deserve it because you are a champion, my friend.
Oh, thank you, my
Keep
friend.
being you. Yeah.
Thank you.
All right, and now we're ready to rumble because this is about how we're gonna save a man one molecule at a time. And I have a story for you, Adam.
All right.
This is about an introduction to a hero, Hartwell, who almost has an insulin overdose.
Yeah.
Basically, our villain wants to see him react so that the algorithm that the villain is creating can learn how to or how not to react to certain challenges in real life. And Dave, the villain, basically wants the hero to deal with the challenges of healing from a trauma, and he intervenes. One time our hero is invited to spend a night with a girl, and things happen. And things happen there, and the next morning, she makes pancakes for Hartwell. And the pancakes come with what? Canadian maple syrup. And that is mixed with a special kind of chemical. That special kind of chemical is a booster. It boosts brain plasticity, and Hartwell eats this with the pancakes. And so he also has type 1 diabetes, and so he uses insulin for breakfast, and about an hour later, he notices that his blood glucose is rapidly dropping faster than ever before. Yeah. What? How come? He's surprised because he just had pancakes with maple syrup an hour ago, right? So he doesn't know what's happening. And the sci-fi explanation is that the brain plasticity boosting compound made such an effect on Hartwell's body that his brain and energy is now being utilized and used to the extent that this was not normal. This was abnormal. Okay. And so this compound basically rewires the brain in a sense that the subconscious is healed from a battle scars it may have had once. And I think it is a fascinating compound, but it taps into the energy reserves of the body, and for our purposes, this will be glucose reserves and glucose itself. So what do you think of this concept? And if you think this is a cool idea, how would you make it? And is there a way to separate the conscious and the subconscious, in your opinion, based on how active they can be or how much sugar they're using in the brain?
Well, I think the physiological explanation to that is that it's a contradiction. Because you can't... It's difficult to have an insulin overdose when you just ingested a large amount of calories, especially within very refined carbs like pancakes and maple syrup. So you'd have to inject a enormous amount of insulin in order to induce hypoglycemia.
Or I would challenge that idea actually, or just have a high amount of insulin sensitivity so that the ones that is being utilized.
Yeah, I mean, it's reasonable to say that this miracle drug somehow increases insulin sensitivity, but that would go for the whole body. For the brain, I would argue it doesn't really matter because the brain metabolizes sugar first.
Mm-hmm.
It can't really eat anything else. It can use ketone bodies, but only if the glycogen and glucose reserves are bottomed out.
Mm-hmm.
So it would always eat the sugar first.
Mm-hmm.
It can't really thrive on anything else like protein or fat or full fat.
Mm-hmm.
Just the ketone bodies, which are very simple molecules.
And the question also was like, how would you make a molecule that basically like specifies a region of your body, namely the brain, and ensures that insulin sensitivity is boosted for the sake of that region being more active in your body, the brain?
Yeah, so I guess there was also a question about what it means. What this means on a molecular level, right?
Yes.
When we say a certain cell type is more sensitive to insulin than something else, it all comes down to the receptors, basically. So what insulin does is that it induces the transport of glucose receptors into the cell membrane.
Mm-hmm.
But there are also insulin receptors on the cell surface. And it is potentially possible to increase the density of insulin receptors on the cell, which would in turn increase its sensitivity to insulin, which in turn would cause it to insert more glucose channels into the membrane, which in turn would make it extract more glucose from the bloodstream. And so hypoglycemia occurs when glucose is depleted from the blood, and that could be quite dangerous. And during an insulin overdose, this is why the blood sugar drops quite drastically. And this is, I guess, why he would be in trouble.
Yeah. Well, Hartwell does not like to get in trouble, but for some reason he gets into trouble all the time.
Yeah, but it's an interesting question if insulin sensitivity can be boosted. I haven't quite read a study that where they did that. The issue with the brain is that it has very low sensitivity to insulin because it's the primary organ that needs to be fed. Mm-hmm. So the reason why hypoglycemia is so dangerous is because
the brain can shut down.
The brain can shut down. If it doesn't get enough sugar, then it enters a state called diabetic ketoacidosis.
Right.
Wherein it's only type of food with the ketone bodies I mentioned previously.
Right.
Which can be depleted easily, and this will essentially induce a standby mode, a very low energy consumption state where most of the functions can't work. So you get fainted, you fall into a coma.
Right.
It's not a pleasant state of affairs. But the funny thing is that brain activity does not necessarily consume that much calories.
Mm-hmm.
If you get what I mean, it's very difficult to fall into a state of hypoglycemia just because you are sitting there and thinking.
Is that 100% confirmed? Because I imagine that this is a scenario actually where Hartwell is going through a situation where the brain is using the most energy in his body, and more than the energy being used by his muscles combined. And this is just a scenario I imagine. I'm not sure if it's real or not, but let's talk about it. So what is the difference between how a neuron, a brain cell uses insulin, and how a muscle cell uses insulin? Why was it believed for many years that the neurons do not even use insulin?
That's a good question. So, again, it all comes down to receptors and the channels. Mm-hmm. The reason why it was believed that the brain has low sensitivity to insulin or not sensitive to insulin at all is because neurons contain a lot of insulin-independent glucose channels. Obviously, because that's the primary organ you need to keep alive. So this is where the most of the blood sugar will go first. Mm-hmm. If it's in excess, then the liver will start metabolizing sugar and build up a lot of glycogen stores, which in a normally functioning individual will begin to get depleted if we turn into a fasted state.
But it doesn't work for Hartwell because Hartwell is type 1 diabetic.
Hartwell has a problem of moving sugar from the blood to the cells.
Okay.
And, the primary hormone that's responsible for that is insulin. Mm-hmm. And since he's type 1 diabetic, his pancreas doesn't work. There's no secretion of insulin. The whole signaling network is compromised. So his blood could be full of very valuable sugars, but his cells, including...
his brain cells and muscle cells?
Not the brain cells, because the brain cells, again, will use the glucose first without any interference from insulin.
Okay.
They do have some sensitivity, as we now know, because we have fMRI studies. Mm-hmm. We can more accurately track the glucose molecules path once they enter the body. And this could be done by a variety of methods, but contrast MRI is probably the best. And this has undergone significant advances in the last few decades. So I suppose it's conceivable that since this compound somehow enhances the insulin sensitivity of the brain, and the brain activity consumes more sugar than the rest of the body, and then that leads to hypoglycemia. But it's just difficult according to our current understanding, to really imagine that the brain would be so active that it depletes all the sugar from the blood first, and to the point where it actually goes into a fasted state. And it gives you hypoglycemia by itself. Very interestingly, to the best of my knowledge, there is no current evidence to suggest that just thinking very hard versus just sitting there, not really thinking about anything, has a difference in energy consumption on the level of the brain cells.
That's interesting. Okay, so I don't know. Really?
So the primary goal is basically just to keep the brain alive.
Okay.
Now, obviously, rewiring of the brain, or plasticity, which generally refers to the ability of neurons to make new connections, which the potential of this is usually higher in children. This is why they are easier to teach than adults, so they learn things like languages more easily, because their brains are more plastic. They can form new connections more readily than in the adult brain, because at the adult evolutionary it has been the case that it's more skill-based and experience-based, not necessarily learning-based. So that has a time during our development, whereas after a certain point you just have to rely on the skills we have already learned and apply them reliably. And this is why adults are better at this than children. Children are still learning.
So we gotta answer the big question, the elephant in the room. Then, I think we gotta answer the question once and for all: can someone have more brain cells than muscle cells?
It's an interesting question. We should probably look that up, because I'm not quite sure how many brain cells we have. And how many muscle cells. I suppose it also depends on...
Well, by comparison, the muscle cells have more mass in our body, right? So your brain is lagging behind. It's a Goliath and David situation when it comes to the muscles and the brain cells by weight. Right. But does that mean that the cells add up? We must find out, and the way we're gonna find out...
They might not, because muscle cells, I think, tend to be heavier than neurons.
Okay.
It's an interesting question, but I'm not convinced that it comes down to the number of cells, or depends on what the outcome you would like to reach.
Okay, so I'm interested in knowing in the end, what uses more insulin and what uses more sugar?
Muscle definitely uses more insulin.
Okay.
And the brain definitely uses more sugar.
Okay, really?
Yeah.
That's how it works?
Yeah, definitely, because the brain has a lot lower sensitivity to insulin, like liver and muscle.
Lower or higher?
Lower, because it metabolizes glucose readily. It doesn't need insulin to ingest glucose from the bloodstream.
Yeah, but that would mean a higher sensitivity.
No.
No?
Because insulin...
so you're saying that the brain has high sensitivity to glucose, but low sensitivity to insulin?
Yes.
How's that work?
It works because there are lots of insulin-independent glucose channels in neurons. And this is why we used to think that the brain is probably insensitive to insulin, or insulin just does not really affect it. As it turns out, that's not quite correct, but it still is the case that insulin primarily affects other organs, like the liver and muscle.
Now I understand it. Okay.
Yeah. So I think there are 4 kinds of glucose receptors called GLUT1 to 4, and I think GLUT4 is the only one that's introduced into the cell membranes by insulin. Okay. The brain has lots of GLUT3, I think, which is completely independent from insulin.
Okay, so
It's just there no
tell
more.
me more about this because I'm interested. So these GLUT receptors, these are receptors?
Yeah, glucose transporter.
Okay.
Yes, that's.
So how do they work? What do they do? And then name the ones you know.
Right. So, to the best of my knowledge, the cell membrane is not readily permeable for glucose. Right. It's a bit of a bigger molecule. It is polar. I suppose cell membranes generally allow they readily transmit fat soluble and small molecules, but once you get to a certain size, you need some sort of facilitation. It usually occurs by channels. I should remember this, but I don't think that glucose is efficiently translocated passively. That means without any expenditure of energy at all. So there are some molecules.
The moment when a glucose arrives to the cell, there's an energy expenditure to open the gate for the glucose?
Not necessarily to open the gate, but to translocate through the membrane.
Okay.
Require.
I don't think I understand the word translocate, so what does that mean?
It just means crossing the membrane.
Okay.
So, in order for glucose to cross the membrane, its transport is usually coupled to a transport of another molecule, like sodium.
Okay.
So, where glucose would go from outside to the inside, then sodium would move from the inside to the outside.
Okay.
And they move down the gradients, ostensibly, since the concentration of glucose in the cytoplasm is usually a lot lower than that in the blood.
Mm-hmm.
But this gradient is usually not sufficient enough to transport glucose in sufficiently faster and in sufficient quantities.
Mm-hmm.
So we have this clever mechanism where it's coupled to the down gradient transport of sodium. It's called antiport because they move in opposite directions.
Mm-hmm.
So when I say energy expenditure,
the antiport is doing what?
It's moving one molecule in one direction and the other in the opposite direction. So basically like that, yeah.
Okay. So is that true that glucose can be kicked out of the cell? Is there an antiport for glucose? Or glucose is always...
Well, it's antiported into the cell, but there is no reason why it should be kicked out of the cell.
Okay, so based on that reasoning, we understand that once the glucose gets into the cell, it's in. And it's in
and it gets metabolized or built into glycogen.
Yeah, so the reason why I find that to be fascinating is because now I'm already imagining the scenario when one sort of candy bar is somehow radioactively linked to some sort of other process, and you can measure how or when that candy bar was eaten, because it would be part of your cells, and then that would become part of your body.
You can do functional experiments like that. You can do isotope labeling of glucose that actually you can do that with humans as well. Mm-hmm. You can isotope label glucose, have your subject eat it, and then you can detect its transport and diffusion through the entire body using fMRI.
Mm-hmm. Pretty amazing sci-fi stuff. So
it's not sci-fi; it's happening as we speak.
Oh, that's even better.
That doesn't say to beyond.
Well, we're gonna go from the future back to where it came from Newton. And Newton studied the mathematics and the limits of mathematics, and came up with a couple useful tools. He was also interested in slicing up time to as little pieces as possible. And now I'm interested in knowing what the shortest time of a cell can live for. How long can a short time be which a cell can live for that amount of time? And perhaps you can give me a reference for this idea. What is a cell cycle like, and when is it the fastest? is there like some sort of scenario when we can facilitate a cell cycle where things happen and things no longer happen for the cell, meaning the cell dies, for example. And if we can do that, what is the shortest amount of time that we can achieve for a cell to live and die?
That's an interesting question. I suppose there is not much evolutionary advantage to a very short lifespan for a cell. In fact, I believe the first cells or proto-cells were thought to be essentially immortal because they couldn't have existed otherwise.
Oh.
And, we use immortalized cell lines in the lab all the time, which means as long as you keep them fed, they will just keep reproducing and until the end of time, until they have something to eat.
Wow.
And are kept under the right conditions. Obviously, if you put them to minus 80 degrees, they'll probably won't be as active as on this very comfortable 35, 36 degrees that we have in your apartment. I don't know why it's always so hot in here, but
it's okay.
Supposed to be good for ourselves. Anyway,
so then I changed my question then, not from being dead to alive or alive to dead, but how, what's the shortest amount of possible cell cycle if we were to...
The shortest time for it to complete one cell cycle. That's actually a very reasonable thing to ask because cell cycle lengths differ from cell type quite widely. And I suppose the shortest division cycles occur during embryogenesis. This is when, under the span of a couple of days, we can go from one cell to hundreds.
Wow.
So the cell cycles of those cells can be, I suppose, just a few hours then. So, just for the uninitiated, cell cycle refers to the process wherein a cell divides, and that's a very methodical and strict process wherein several checkpoints need to be fulfilled. And during that cycle, a genome needs to be duplicated with sufficiently high fidelity, and then we come to the stage of mitosis, wherein one cell becomes two, and then we can start the whole thing again.
And it can do it again, as long as it's well fed.
Yeah, some cells can do it indefinitely. Most cells in our body are not like that. It's the average number of divisions are thought to be around 50. 50? Yeah.
Wow. Average. What is a? That's a good question. What does an average cell look like in our body? Is it?
I don't think there is such a thing as an average cell.
In an average day, in an average cell's life,
the cell that's probably present in the most numbers of the squamous tissue of the skin.
Okay.
It's our largest organ. So skin cells are a very well-defined structure, but they differ widely from, say, a white blood cell or a neuron or a muscle cell. Cells in the human body are highly specialized structures.
I have another question for you, which is about data that we may not have. Okay? So I'd like to ask you, and we may have to pull out a calculator for this, but I want to know, and we can skip this if you don't know the answer, but I trust that we would find something if we put their heads together. And I think it's interesting. So how many molecules of insulin does one muscle cell require to fulfill the shortest possible cell cycle? And if we don't know the numbers, how would you go about trying to answering that?
Yeah, that's interesting.
Because I can give you numbers later.
Yeah. The lifespan of a muscle cell depends on quite a few things. Most importantly, is it fed enough? What we see during anorexia nervosa or very long-term fasting is that muscle cells begin to die as they start cannibalizing each other. That's probably the most critical state of being fasted or starvation. that we see in the bodies where proteins will get started to metabolize. That's one of the worst conditions you can imagine for a cell, muscle cell especially. Muscle cells obviously have a certain sensitivity to insulin. We store a lot of glycogen in muscles, which can be depleted quite quickly during strenuous exercise. But I don't think you can give a very concrete answer as to how many molecules of insulin you need to supply to a muscle cell in order for it to have the longest possible cell cycle. I'm not quite sure about the normal lifespan of a muscle cell, but that's probably the longest time that it can survive for, which is where it's completely happy and fed all the time. But I think that depends primarily on the availability of the fuel, not necessarily insulin. You need insulin to get the fuel from the blood to the muscle cell. But if there is no fuel, then the muscle cell will eventually start to die.
Mm-hmm. That makes sense.
And you need as many molecules of insulin that you need to achieve that.
Makes sense.
A single molecule of insulin can insert quite a large number of glucose channels into the
Yeah, because an insulin molecule is big. It's if you were to take 1 mole, which is a standard unit in chemistry, 1 mole of insulin molecule, it would be about 5 kilos.
Yeah, it's a peptide, I believe. It's around 20 amino acids. I'm not quite sure.
Well, there are different insulins being prepared in factories these days, but the ones I checked out was Lispro, and I have data on this. So how does the math work out? Let me tell you. I got some data for you. In an insulin pen I use, there are 3 milliliters of insulin solutions. Okay? On the box it says 1 milliliter has 3.5 milligram Lispro insulin compound dissolved. This is good for me for about 7 days. And so if I calculate it, it is 10.5 milligrams of Lispro insulin plus my long-acting insulin that can sustain my body. But we will focus on the fast-acting now. So we have about 1.9 times 10 to the power of 18 molecules per week of fast-acting insulin.
Yeah.
And if I divide the number of seconds we have in a week, then I get that basically like I get how much insulin would I need every second.
Yeah.
So I make that division, and I need approximately 1.8 times 10 to the power of 12 molecules of Lispro every second. So that's a lot of molecules per second for a body, I think. But the interesting thing happens at the end of the calculation, so let's continue this. To the best of my estimation, I have roughly about 37 trillion cells, 3.7 times 10 to the power of 13 cells. If you divide the number of Lispro molecules per second by the number of cells, you find out that each cell would need approximately, you guessed it, 0.05 molecules of Lispro per second. And this value is less than 1, meaning not every cell would need a full molecule of Lispro each second. Only a small fraction would. Yeah, roughly 5 in 100, in every 100. And the total cells would require basically this analogous great coordination of which cell needs what. So I'm basically, again, back to the insulin sensitivity question, the more sensitive cell will require or will hopefully use the insulin faster, right? Yeah. And I'm interested in, like, on a molecular level, what can boost the insulin sensitivity? I know we talked about this before, but tell me more about it.
Yeah, so again, it's basically the number of insulin receptors per cell.
Mm-hmm. Can there be more? Can we manufacture cells to have more?
Yeah, of course. We can genetically manipulate cells, and that's probably the easiest way of doing that. I don't necessarily know of a hormone that can increase the number of insulin receptors in the cell. There might be one. There might be a way to exogenously introduce that. But I think the number of insulin receptors per cell is established during cell division or cell differentiation, to be more exact. Because there are so many tissues and the insulin sensitivity needs to be set to a very particular amount. And that numbers are basically established during embryogenesis when the separate tissues form and they become looking different from each other.
Does it surprise you at all? Because for me, when I did the calculation and came up with this, that not every second is gone past in this world where I need insulin in all my cells. That surprised me. Because the basic narrative of people with type 1 diabetes is that we always need insulin and every cell needs insulin. And then the new narrative I'm hoping to create is that not every cell needs insulin all the time. Matter of fact, you don't need anything all the time. You need things at the right time.
Yeah, it does not surprise me at all. Because I think there is an important lesson here in data science, but also in life sciences, which is the dangers of averaging out everything. Especially in the context of a living organism, the requirements and the circumstances in which a hormone is released can change quite rapidly and widely with respect to time, both space and time. So if you just take an average over time, you will end up with a number that does not really reflect real physiology. Just as we found, it's basically less than 0.05 molecules per cell per insulin per second, if I understand correctly. That doesn't really tell us much about how cells work, does it? Because you don't need insulin for every cell in every second, because it all depends on the amount of glucose in the blood, which is not always constant.
Mm-hmm.
You don't have to transport glucose from the blood to the cells all the time, even if you are type 1 diabetic. And not every cell needs the same amount of insulin. So there could be cells that don't require insulin at all, and others are completely dependent on it.
Right.
It depends on the context, and that average won't really reflect these important differences.
Yeah, and it always changes, right? So it can change a lot based on the status of each cell.
You mean the requirement for insulin, or? Well,
I'm saying that if I go for a run, the whole body moves. The whole body makes a shift in my physiology, and my physiology dictates what my insulin sensitivity is. And if my insulin sensitivity changes, I assume my sugar receptors change too, and I become more receptive to those. So it's a
come to think of it, there has been some data published about this that says that insulin sensitivity essentially increases during strenuous exercise, which I think you introduced me to because I wasn't aware of it before. And essentially how most hormones work, not insulin, but the hormones that would be released during strenuous exercise, like noradrenaline or cortisol or things like that we think of as stimulants, that cause a breakdown in glycogen and release of sugar into the blood and then into the cells, because those are cells that are working, they'll keep you running, and then they need to be fed. And it's entirely possible that by some mechanism, these hormones affect the number of insulin receptors, affect and increase the number of insulin receptors into the cell membrane. And they do that by modulating gene expression, which is what most hormones do. Insulin is considered a hormone, but its primary function is not thought as bringing about the change in gene expression.
Okay.
It could. I mean, I suppose.
Well, the way I think about it is that it's a hormone for energy. I don't know if I'm right or wrong about that, but I want to know more now about like what it means to have a hormone for energy, or like, yeah, because this is how I think about it. Do you think I'm wrong, or how wrong can I be?
It depends on what you mean by this.
Well, if I don't use insulin as a type 1 diabetic, I feel less energetic. And if I overuse it, even sports people back in the day have used it as a stimulant, as a doping, kind of like doping thing. But I call it a hormone for energy. How does energy work? I know about ATP and ADP and all these things, but I don't put the puzzle pieces together just yet. So how does energy work, and where does insulin and glucose come into the picture?
Yeah, biology is not very concrete like that. And it's easier to think in terms of fuels rather than energy. Because energy in biology derives from a variety of sources.
Mm-hmm.
One of them is the burning of fuels like glucose, but we can use other sources of fuel like fats and as a very last resort, proteins.
Mm-hmm.
Insulin is very important, but it only covers just one sort of piece of this whole puzzle, which is to transport fuel from one compartment into another.
Mm-hmm.
And that's a very important step because that fuel has to be metabolized at the right place.
Mm-hmm.
And at the end of glucose metabolism, what we get is ATP, adenosine triphosphate, which is thought as the energy currency of the cell.
Mm-hmm.
So if the cell needs a rapid release of free energy, then the molecule it will turn to will be ATP. And that's generally thought of as the currency in which energy intensive processes are paid for. So say that you need to push something out of the cell that would go against this concentration gradient, which is an energy intensive process. It requires an investment of energy. Now that transport process would require the hydrolysis of ATP molecules. ATP is said to have a string of high energy phosphate bonds. Not the best way of putting it, but what basically happens is then when those phosphoanhydride bonds get hydrolyzed, there is a release of energy. And that could be input into mechanical energy that would expel those molecules out of the cell.
Mm-hmm.
But it could be used for a wide variety of purposes. Muscle cells use ATP for contracting.
Okay. Yeah.
And they get that ATP by metabolizing glucose, making glucose from glycogen, and whatnot. So I don't know how accurate it is to call insulin energy hormone. I would call it a fuel transport hormone because it helps moving the fuel from one compartment into another.
Makes sense.
Yeah. And that fuel happens to be the most important.
Say that again. How'd you call it? A mover? A transporter? A transport what?
A fuel transport hormone.
Fuel transport hormone. I like that. That's romantic.
Yeah. I don't know why, but whatever floats your boat.
Awesome. So we have been working on cool stuff. Let's just call it cool stuff. This podcast being one of them. And we have a previous written conversation about how a movie star, Wolverine, Hugh Jackman, and another movie star, Ryan Reynolds, Deadpool, survived the end of Deadpool and Wolverine, the movie. We have seen the movie together. No need to comment on how good or bad the movie is, but we sure have thought about it, the ending. And yeah.
Let's just say that our discussion of the ending was probably much more intriguing than the movie. Let's just leave it at that.
Hey, I like the movie. Adam is critical of the movie, but hey, I like the movie a lot. So yeah, I've seen it multiple times. But yeah, so it's just like always fascinating to like have a friend like you, Adam, to basically have have a ping pong racket in his hand and say, "I'll play ping pong with you whenever you have a crazy idea." So I'm honored and flattered to call you my friend, and yeah, it's just like hopefully we're gonna keep doing this for a couple more decades.
Yeah, I hope it will. You know, I don't really think of myself as being above or below you. You know, we are friends. We just have these conversations because we like to have them.